Hi, my name is Karen Dietz, and this is my partner Nathaniel Cisneros. We're going to be doing a presentation over the evolution of stickleback fish and the presence versus absence of their pelvic structure. I'll start off by giving you an overview of the experiment. We're going to be conducting 3 experiments in one. Each of these will analyze the evolution of stickleback fish. Experiment 1 is analyze fish from lakes. In this experiment, we're analyzing the pelvic structure of stickleback fish from 2 different lakes. And we will use our data from this to draw conclusions about the possible environmental factors affecting the evolution of pelvis morphology. Experiment 2, analyze fossil fish. We're going to characterize the pelvic structure of fossil stickleback fish, and we're going to estimate the rate of evolution of pelvic structures in these populations. For experiment 3, pelvic asymmetry, we're going to analyze the reduced pelvis trait in more detail by scoring sticklebacks from 2 freshwater populations according to whether their pelvis is larger on the left or on the right, making them left bias or right bias.
Allow me to give you some background research on our experiments. Well, evolution is when species undergo massive evolutionary changes. They begin to change and adapt to their environments. This exact cause is still unknown to scientists even to this day, even though we may have a few good guesses on why this happens. Because even though evolution occurs to when species evolve over time, we also see that species modify, such as gain or lose limbs in the experience. As you can see, for example, in our picture right there, you see 2 fishes, which is contrary to which is in comparison to the experiments we have today. You have one type of fish that has spines on its back, which it has an absence of pelvic structure, but you have another that has the pelvic spine, but it does not have spines on its back. It instead has armor plating. Stickleback fish are not the only species to have gone this loss or gain of pelvic hind limbs over time. Whales, manatees, and some former reptiles have undergone it as well. Many members of this Gastrus aculis, or stickleback fish, that's the scientific name for it, have the presence of this marine pelvis. It allows them to deter possible predators, basically. However, around more than 2 dozen geographically isolated populations completely lack or have an incomplete version of this pelvic structure. How does this happen and what does this mean? Scientists begin to believe that morphological differences among these populations of stickleback can be attributed to 1, geographic differences, 2, genetic mutations, 3, adaptation, or 4, just natural selection on a species environment. To find these genetic mutations responsible for this gain or loss in pelvic structure, scientists actually analyze chromosomal differences in the region that codes for this pelvic structure in both marine and freshwater populations of the species. Turns out that this primary gene is pituitary homobox transcription factor, or PITX1, is the gene responsible for this pituitary development. While sequencing and expression studies has shown that the gene in freshwater fish encodes for a normal protein, but however, it is found to be failed in expression to in a certain pelvic region, suggesting a regulatory change, which is basically why scientists, after running SNP, which is a form of genetic sequencing in this region, they have found that many deletion mutations, meaning that this PEL gene rendered the PIX gene basically immobile and mute. Evolution can occur in a multitude of ways, from natural selection, gene flow, change over time, genetic drift, etc. As a result, traits can evolve rapidly or slowly, differentially, leading to the establishment of many different diversity of species over time, each with a different structure, different genetic variation, different morphologies, etc. But more often than not, evolution can cause traits to undergo repeated evolution, which is basically the potential loss or gain of new development, morphological or structural changes. A great example of this is, of course, pelvic time bone in stickleback fish. Scientists have seen that the loss of this has been referred to deletion mutations within the PIX1 gene, as I said before. This basically what happens is that one major cause of this phenomenon can either be attributed to one, high mutation rates, or two, DNA fragility. DNA fragility is when genomes regions are more likely to experience mutations and deletions than other certain parts of the genome. Scientists summarize that DNA fragility and high mutation rates influence the genomic pathways of adaptive evolution, basically.
Our hypothesis is certain features in these environments, Coyote Lake and Bear Paw Lake, are causing morphological evolutionary changes in the pelvic structure of different stickleback fish populations. Here we're going to give you some of our methods.
These are our variables. Our independent variable for experiment 1 is Bear Paw Lake and Frog Lake. We are getting the fish from this lake. This is the variable we do not change. And our dependent variable is pelvis morphology. This is the variable we are looking to measure, or we are looking to see a change in between populations of these two lakes. Our independent variable for experiment 2 is fossil position. We are looking for fossils in positions based on layers of sediments in the earth. And our dependent variable is our type of fossils. We are measuring the difference and the variation in each type of fossil found in each position of layer. And our experiment 3 independent variable is Bear Paw Lake and Coyote Lake. You don't change an independent variable. Independent variable is where it's the variable that cannot be changed. We cannot change the fish, or we cannot change the lake. We are getting fish from both these lakes. And our dependent variable is our pelvic spine. We are measuring the differences between left and right bias in our pelvic spine. Our control variable, or the one we basically established, is measuring the changes in stickleback fish with all possible variables. This is our control.
For experiment 1, analyze fish from the lake, we did it in 3 different parts. Part 1 is staining the fish. So we're going to treat the stickleback fish collected from Bear Paw Lake in a solution that'll basically dye the bones red and make it much more easier to identify. And part 2 is scoring the fish. We're going to categorize each specimen according to whether it has complete, reduced, or absent pelvis. For part 3, we'll be graphing our data. As you can see, Bear Paw Lake had all of its fish that were observed either in reduced or absent for its pelvic structure, as opposed to Frog Lake, who had all 20 complete. Experiment 2, analyze fish fossils, again in 3 steps. Part 1, we are preparing the fossil. Since it is a fossil and it's not a fish that we can stain, we are just going to collect the slabs of rocks that were brought to the lab, and we're going to carefully uncover and stabilize them by repeatedly spraying them with acrylic spray to prevent deterioration. Part 2 is scoring the fossils. Now that the fossils are prepared, we will categorize the fish according to whether they have complete, reduced, or absent pelvis. And part 3 is our data again. You can see layer 1, layer 2, layer 3, 4, 5, and 6. And you can see how throughout the layers they vary from different complete, reduced, or absent structures. Experiment 3, pelvic asymmetry. Part 1, again, is staining the fish, just like we did for our first experiment. Before we can determine the asymmetry, we need to prepare the fish by staining them with the exact same solution so that we can see their bones more clearly. Part 2 is scoring the pelvic asymmetry. We're going to analyze the reduced pelvic trait in more detail by scoring sticklebacks from 2 freshwater populations according to whether they have a larger pelvis on the left side or on the right side, making them either left bias or right bias. Then part 3 is our data again. You can see that in Bear Paw Lake and Coyote Lake, the majority of the fish will have left bias.
And here are the results for our first experiment, analyzing fish from both lakes. Out of 40 fish, 20 from Bear Paw Lake and 20 from Frog Lake, we see that the 20 from Frog Lake have complete phenotypes, complete pelvic phenotypes, which means the pelvic bone within their system is present and alive. Basically, it means that Frog Lake is connected via a small stream. It's not geographically isolated. There's no evolution really occurring here, and there must be a lot of predators here because stickleback fish need their pelvic structure to repel against certain predators. In contrast, compared to Frog Lake, we have Bear Paw Lake, which we have 20 individuals from. And note this about Bear Paw Lake, it is completely enclosed and there is not as many predatory fish. If not, there is none at all. And as we can see here from our phenotype totals, we see that 16 fish out of 20 exhibit a phenotype for reduced pelvic structure, and 4 completely have a phenotype that is absent. What does this mean? This means that the fish that are isolated within Bear Paw Lake have become genetically different from the ones in Frog Lake. They don't need their pelvic structure. It has basically evolved out. It has basically been, it's in the process of being lost through like evolution. These are results from experiment 2. As we can see here, we have 5 different layers. Layer 1 being the oldest and layer 5 being the youngest, the earliest, the topsoil. As we can see here, as we go down into the furthest layer, we see that most, if not all, the fossils exhibit complete phenotype structures for pelvic backbones among sticklefish. This means that originally the population had complete phenotypes for the stickleback pelvic structure. But as you go further up the layers, you start to see that the complete phenotype gets reduced and even lost among layer 5 and layer 6 of the sediments. But you see a substantial rise in reduced and absent phenotypes in accordance to the loss of complete phenotypes. What does this mean? That means that stickleback fish pelvic structure adapted to its environment, either be of genetic drift, either it be just because of natural selection or due to lack of predators or genetic isolation. We just know that this caused one of these factors, or maybe all of these factors, caused adaptation in its environment. Results 3. Here is the experiment results for our pelvic asymmetry experiment. Out of the 40 different fish, 20 from Bear Paw Lake and 20 from Frog Lake, out of all of them, we had 37 measure for left pelvic bias and 3 measure for right bias. We can see that there's basically a massive pelvic asymmetry towards left bias because that is what keeps most of the fish alive. That is what keeps the most of the fish genetically stable. That is basically evolution. And the right bias seems to be sort of evolution away from, left bias.
In conclusion, I'm going to go over our hypothesis again. So the hypothesis that we had made was certain features in these environments, Coyote Lake and Bear Paw Lake, are causing morphological evolutionary changes in the pelvic structure of different stickleback fish populations. Was our hypothesis correct? Yes, it was. After performing 3 experiments, we observed reduction in the pelvic structure of stickleback fish populations that have been trapped in different lakes after access to the ocean was cut off. So basically, there was just not a stream that would allow them to go from freshwater to ocean water. And they were isolated, so there's no predators in these environments anymore. Why does this happen? The pelvic structure in stickleback fish was reduced or found completely absent because of factors contributing from a change of residency where there is no longer a primary predator. As opposed to being in the open ocean where you see all sorts of different species that can obviously eat you, eat me, eat you, eat these little fish. There is not one anymore where they are secluded. How does this happen? Through many years, the work of evolution and natural selection has evolved populations of the stickleback fish. An explanation for this is the structure and spines of the stickleback pelvis are used as a defense mechanism against its predators. When the fish are relocated to a new environment where there are no predators, the pelvic spines become useless.
Broader impacts. Why does this experiment's results affect the real world? How does it affect the real world? How does it affect us? Well, the results of our experiment show how evolution is not only influenced and affected by natural selection, but by a multitude of environmental and genetic differences among Big Bear and Frog Lake. Basically, they give rise to substantial genetic differences between these populations of stickleback fish, where Big Bear natives exhibit complete and whole pelvic phenotypes, and those in Frog Lake complete exhibit phenotypes that are absent or incomplete, basically. Evidence for this is also increasingly present within the fossils of ancient organisms. Charles Darwin, father of evolution and founder of, the theory of evolution, basically said one day himself that evolution is described as descent with modification. Early evidence of evolution revolutionized our traditional beliefs that, species and humanity were on on basically higher levels of speciation, that life didn't change, everything was perfect, life itself was basically perfect and in nirvana. No, that's not true. Life is always constantly changing through adaptation, natural selection, other various factors. From our experiments and our data results, we can confirm that the effect of environmental factors can have on a particular species, for example, our stickleback fish, can completely affect and transform a species' genetic variation or morphology, not necessarily in their structural morphology and development, but also in their genome, as shown earlier. Their structural development and their overall identity can give rise to independently and almost a new species. The strength in this experiment leads to the methods that we used in our three different experiments, which all more or less gave the same outcome, that evolution is present and these fish are evolving based on their environment. Analyzing the set of 40 fish from each different lake, both of them, provides a small present day example that there is differentiation in these pelvic phenotypes, and that each of these phenotypes is different from the other based on their environment and based on their surroundings. Fossil records also show that the genetic variation between um, in pelvic bones of stickleback fish has changed over time, meaning it's not just environmental factors, it's also change over time. As we saw in layer 1 through 5, layer 1 had complete phenotypes all the way, while in layer 5, incomplete or absent phenotypes dramatically overtook complete, which were basically non-existent. Also, lastly, it shows that there's descent with modification over time, because also a massive majority of our recorded fish were left by us not right. This shows that based on our findings, scientists and other researchers can use our data as a representation of geographically isolated non-predation and little changing habitats isolated, and the effects on those species compared to ones that are open, massive, full of diversity, and full of predators. For an example, our experiments show that how evolution can occur in small environmental differences as opposed to millions of years. This is one example of how evolution can occur over millions of years. This diagram right here, which we've included, is a modern day ancestral tree of elephants, both Asian and African, the extinct woolly mammoth, and all their common ancestors and their genetic and ancestral lineage. Thanks to modern technology, such as radiometric dating, fossils, genetic testing, genetic analysis, we can finally look at genetic lineages and evolution of the past. As you can see right here, the y-axis right here represents millions of years. And as you can see, these species evolved from a common ancestor over millions of years. As you can see right here, our present day species are, of course, Asian and African elephants. But their most closely related ancestor is the woolly mammoth, which is unfortunately extinct. But as you can see right here, you see that all of these organisms, all of these species are in fact related to one another based on the common ancestor, where which each species branches off. Understanding how life fundamentally evolves, changes, and adapts over time has not only allowed us to fundamentally understand how Earth, how animals have adapted, how life has adapted and changed over itself, it has increased our knowledge of genetics, our scientific knowledge of the natural world. This can greatly benefit our understanding of animal lineages, genetics of animals, our genetics, where we come from, our evolutionary history, animal biology, medicine, climate change, even mass extinctions, and it better understands how we view life now.
This is just a page basically with our references. Thank you for watching our presentation over stickleback fish and the evolution of their pelvic morphology.